Welcome to the wonderful world of biology, where all your sciencey questions will be answered. Let's talk about dogma, baby. Let's talk about you and me. Let's talk about polymerase, DNA, and mRNA. That may be. Let's talk about dogma. Let's talk about dogma. Oh, yeah. Zooms off into the distance. What is central dogma? Central dogma is the process by which instructions of DNA are converted into a functional product. It was first proposed in 1958 by Francis Crick, the discoverer of structure of proteins. Central dogma provides the basic framework of how genetic information flows from DNA sequences to a protein product inside cells. This process of genetic information flows from DNA to RNA to proteins, and this is called gene expression. There's 3 steps to central dogma, which is replication, transcription, and translation, and you'll find out more about these 3 steps if you keep watching this video.
DNA replication, or the copying of cells. DNA has nearly 65 billion base pairs of DNA in your genome. They all must be copied correctly when any of the trillions of cells divide. DNA replication is semi-conservative, meaning that each strand in the DNA double helix acts as a template for the new complementary strand. First, you start with your DNA double helix. Then you get your hydrogen bonds and they break and open up the helix. Then each strand of DNA acts as a template for synthesis of a new complementary strand. And finally, replication produces 2 identical DNA double helixes, each with 1 new and 1 old strand. And the third part of central dogma is translation. So what happens with translation? What happens is that the mRNA has a DNA message and the tRNA helps interpret the mRNA, and this occurs in 3 sites in the ribosomes. The first site is the A site. It holds the AAs. The second site is the P site. It holds the polypeptide chain, and then the E site, or the exit site. There are 2 different types of ribosomes where translation happens. One is the free ribosome, where synthesized proteins stay in the cytosol and functions there. And the second one is the bound ribosomes. This is what takes it to the ER. They make proteins and of endomembrane system and proteins for secretion. The process. First step is initiation. Small subunits bind to start codons on mRNA, and the tRNA carries METs. That attaches to the P site, and then the large subunits also join and attach onto it. Then the second process is elongation. First is the code recognition. This is when tRNA anticodons matches codons in A site. Then peptide bond formation happens, where AA in the A site forms bond with polypeptide in the P site. Then there's the translocation. And this is when, after tRNA is in the P site, the tRNA moves to the E site and exits the ribosomes. This process happens over and over and over again. This is redone over and over again. Then the third step and last is the termination. As soon as it starts, it gets all the way down to stop codons, which is the UAG, the UAA and all that, and it reach a translation to stop it. Then a release factor binds to the stop codon. The polypeptide that is formed, the polypeptide chain that is formed, is released. And then the ribosome subunits then dissociate, and everything goes back to normal. What happens to the polypeptide? The protons fold. They are folded and are relocated. How the protein folds is that during synthesis, polypeptide spontaneously folds and coils. And then chaperonin is a protein that helps polypeptide fold correctly, which that helps it spontaneously, quote unquote, form and bond and everything. Then after that, a signal peptide, or a 28A, at the leading end of the polypeptide chain that is occurred, determines destination, and it takes it to the location it needs to be. Then the signal recognition particle, or SRP, brings the ribosomes to the ER. And that's the last process of which central dogma happened.